This provides a good location for monitoring. Our entire system has such a layout. The first step is for our temperature sensors to collect data on the thermal patterns in the breast tissue and the rhythmic changes in these patterns. This data is transmitted via Bluetooth to our mobile phones, which then send it to a cloud server. Within the cloud server, we have deployed various machine learning algorithms that categorize and analyze this data. The analysis report generated by these algorithms will determine whether the patient's breast tissue is in a low-risk or high-risk state for breast cancer. Based on the detection results, a report is produced to inform the patient if further medical evaluation and treatment are needed promptly. This entire system has already undergone a series of preliminary clinical trials conducted by our colleagues in the Shanghai branch's relevant departments. The clinical trial was designed by our CRO organization and has obtained approval from the CFDA. Several key points of this clinical report are as follows. 1. It is a multi-center trial. 2. It is not a randomized test, rather an exploratory experiment. 3. It is a double-blind study. The trial was primarily conducted in Shanghai, along with 2 other hospitals. A total of 211 patients were included in the study and categorized into 3 groups. 70 patients were from a normal control group. 100 patients had malignant tumors. 30 patients had benign tumors. The differentiation between benign and malignant tumors was based on imaging studies, specifically using the BIRADS classification system. Patients classified as BIRADS 1 and 2 were considered benign, while those classified as 3, 4, or 5 were considered malignant. After grouping the patients, they were monitored with our equipment for 1 night, totaling 8 hours. The data collected during this monitoring period was then analyzed. After collecting and analyzing the data, we obtained the aforementioned curve graphs. Finally, we will compare our collected data with the data from tissue biopsies to test the accuracy of our results. This process outlines the design of our entire experiment. If anyone is interested, there will be related documents available for further detailed review. In our results report, some key data points were obtained for examination. Firstly, our system demonstrated a sensitivity of 97.7% and an accuracy of 85.9%. Accuracy in this context refers to the true positive and negative results compared with the true positive and negative results from biopsy samples. Of particular concern to us is the false negative rate. False negatives indicate cases where the test indicates the absence of a condition when it is actually present, leading to a misdiagnosis, which is a serious issue. Therefore, during system setup, our aim was to minimize the false negative ratio, even if it meant a slightly higher ratio. This slide shows some references to the background knowledge I just introduced. If you are interested, you can also refer to it. The above is all I want to introduce today. If you have any questions, please feel free to ask them on site or remotely, and we can discuss or share insights together.